Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Sina Sekheche, and I'm not alone. I am with Vedika Maharaj. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. Right, so today we will be doing our Life Sciences Paper 2 revision. But before you start with the revision, let's check what are the topics that you need to study for paper 2. Right, I hope you wrote your paper 1 today. I hope you had a blast with that paper and you enjoyed yesterday's revision. But today, let's look at what you need to study or the topics that are left for you to study for today. Right, so the first topic that you need to study is the topic that you did right in term 1. So you might have forgotten, so you need a lot of revision on this one because you did it a long time ago, right at the beginning of the year when you were still trying to find your footing. So that topic is DNA, the code of life. So you did this for 2 weeks and look at the weighting. Now, this is the weighting, the total weighting of the topic from the entire paper. So how many marks are you expected to score for this topic in your paper 2? It will comprise of 27 marks. Where do you get all of this information? You're going to get this information from your exam guideline, which I hope all of you have by now. Then the second topic that you did that you need to study for paper 2 is meiosis, right? This also you did in term 1. Then you did this with your teacher for 1.5 weeks, and it also amounts to about 14% the total weighting of the question or from the entire paper from section A to section B. Then you have 21 marks that you need to score on this topic. Then for term 1 and 2, right towards the end of term 1, you started with genetics and inheritance, and then you continued with this topic into term 2. So you did this for about 3.5 weeks, and then the total weighting of this is 32%. So it's getting bigger. So genetics, you need to study a lot because then if you check the weighting of this topic, it's 32% of your entire paper. The marks that you need to score for this topic, because it has a lot of subtopics in it, it's 48 marks. So out of this 48 marks, you must set a goal for yourself, how much you need to score on this particular topic. Then the last topic that you need to study for the paper 2 is evolution. This is what you've just done now. It is recent and the information is still new and fresh in your mind. Then you did this for entire 4 weeks now. The time that you did, it tells you the weight also. So the weight on the paper will be 36%, and then the total marks is 54. Now, out of this 54, you must make sure that you score. Then the entire paper for paper 1 will be out of 150, and then all the weightings totals to 100%. So please make sure that you are ready for this. These are the topics that you need to study, and all of this information comes from your exam guideline. So please make sure that you study that exam guideline and you know how to use it. Right. So the paper, how is the paper structured? You may have previous question papers that may be structured differently from the one that you're going to get tomorrow on Monday, or the one that you received today. So the ones that we have now, this is the structure, the new structure for paper 2 life sciences. The paper will still be out of 150 marks. You will still write for 2.5 hours. Then the paper is going to have section A and section B. Only, we no longer have section C, so only section A and section B. So what type of questions can you expect in section A? You can expect to get your short answer question, your objective questions, which are your multiple choice questions, terminology, and the matching column or the matching type of questions. Then the total mark for section A is 50. So this 50 amounts to 33.3% of the paper or of the entire paper. So with section A only, if you aim to get the whole 50 marks for section A, you push yourself at a higher chance of already passing the paper, because if you get that, you are already on level 2. You are at 33.3%. Then section B, this is now a question that has a variety question types. It has 2 questions of 40. So it amounts to 80. So 80, that 80 will be 80 plus the 50, then it gives you that 150. But now These two questions are divided into two questions of 40 marks each, which are divided into 3 to 4 sub-questions. So this is where you're going to find your 2.1, 2.2, 2.3, up to 2.4 maybe. And then you get your question 3.1, 3.2, 3. So they are divided into these sub-questions, and then you find that your question 3 will be out of 40, and then your question 2 will be out of 40 as well. Then it amounts to 66.6% of the entire paper. So if you already scored 33.3% on that section A, then you can still score, let's say you score 40% out of the 60. You already had a better chance of passing this paper. So please expect the questions in this format. We no longer have section C. Right. So what are the things that you need to have, or that you need to be in possession of at this moment when you go and tackle this preparation for paper 2? The first thing that you need to have is Mind the Gap. The Life Sciences Mind the Gap. You need to have the SIP material, and then the DBE study guides. All of this was shared to you throughout the year. And then another resource that you need are your teacher notes. Whatever that you've been doing throughout the course of the year, you need those notes and the activities, because those activities, they have feedback. They told you where you went wrong and where you went right. And the previous question papers. The question papers will be your NSC plus prelim question papers, also the question papers from other provinces, which I think your teacher must have provided you with those question papers by now. If not, you can still access those from the internet, and there are so many links that you can access the previous question papers. Then, most importantly, this is very, very important. All of these are important, but now this is the most important: your exam guidelines and your textbooks. Now, your exam guideline is what is going to give you the scope, or how much you should know per topic. So what are the things that the examiner is going to ask you? So you need to be able to have your exam guideline. Then you can also go to www.education.gov.za to get some of the resources, the previous question papers and other resources that you can source from the education government website. So please make sure that you access this and you have all of this in order for you to prepare for paper 2. Right. So when we mark, when you write, you must have the marking principles in your head, because some of you make mistakes that can be avoided. Now, when you check the marking guideline of every question paper, they have some rules that you need to abide by. So let's check some of the rules that make learners to lose marks. So what are those things? Firstly, if you are asked to give 3 reasons, and then you give 5, only the first 3 are going to be marked. And you may find that your last 2 are the ones that are correct. Then you put yourself at risk of losing those marks. So please make sure that you only write what is required. 3 reasons, only write 3 reasons, because the marking guideline says we're only going to mark the first 3. Then, if tabulation is required, but paragraphs are given, you are going to lose the marks for tabulation. Remember, when it says tabulate the differences between DNA and RNA, you are going to get that 1 mark for T, just for the table, for drawing the table before you even come to differentiating. Then, if flow diagrams are given instead of a description, marks will be lost. If, let's say, you are asked to give a description of how DNA replication takes place, and then you give the answer in a form of a diagram or in a form of a schematic representation, you are going to lose marks, because then you were supposed to describe. So please take note of the verbs that are used when asked questions. Just be sure what you are asked to do. Then, if only the letter is asked for, but only the name is given, or vice versa, then the candidate is going to lose marks. There are questions that will be asking you, give the name and the letter of the part that does 1, 2, 3, 1, 2, 3. Then you're supposed to say A, which is your mRNA or whatever. So you're going to get 2 marks. So if you do not do this, you don't write the letter or you don't write the name, you are going to lose marks. So please make sure that you read your questions carefully and you answer accordingly, because you lose marks unnecessarily. Right. So now we're going to start with the topics. Remember, I said the first topic that you started with was DNA, the code of life, which comprises of 27 marks. So let us take a look at this questions or the DNA. Let's check. What are the possible questions that can come or that you can expect from DNA, the code of life? You can expect it to appear in terminology, interpretation of diagrams, tabulation, description, and calculation. So you must be prepared when you go through your question papers. Just check the type of questions they ask for that particular topic that you are studying. Then you can expect those. Remember, we said the marks are 28 marks. Then for meiosis, you can also expect meiosis in your terminology. Terminology plays a very important part. This is your question number 1.2 of every question paper in life sciences. Diagram interpretation, the description of crossing over, tabulation of differences between meiosis 1 and 2, then the differences between meiosis and mitosis. So these are the things that we are going to cover today, right? So DNA and RNA. From your exam guideline, this is what it says. This is what it says you need to be able to know. So let us check. For DNA and RNA, you need to be able to know what is the structure of DNA, what is the location, and the function of DNA and RNA. This is according to your exam guideline. Then you must know that DNA forms the genes which carry the hereditary information, and then also DNA carries the code for proteins. These are the functions of DNA, and it also plays a role in protein synthesis. No, RNA. RNA is the one that plays a role in protein synthesis because we need the RNA to bring about the required amino acid. Right. So DNA replication and its significance. So you also need to know what is DNA replication and what is its significance. Now, the DNA replication says the DNA makes copies of itself so that it can be shared between the resulting daughter cells during cell division. So we need to have more copies of DNA. So the process that allows us to have that is DNA replication. Then you also need to know DNA profiling. What is DNA profiling? The uses, what do we use DNA profiling for? And interpretation of DNA profiles. You need to be able to interpret the DNA profiles. Remember, you're going to get them in the form of a profile, in a form of a diagram that has bars, the black bands. So you must be able to interpret that type of a diagram. Now, each person has a unique DNA profile. You must know that. What is a profile now? A profile is represented as an arrangement of black bars representing the DNA fragments of a person. So when you get that profile is when those black bars are arranged, and then it represents what? It represents the fragments of DNA of a particular person. Then with DNA and RNA, you must also know what is protein synthesis. Now, process of manufacturing proteins. Process by which proteins are made in each cell. That is a definition of what protein synthesis is. Then you must also know that made in each cell of an organism to form what we form. Remember, enzymes are proteins, hormones are proteins, and new structures for cells. Now, this process is divided into two sub-processes that you need to know and master. The first stage of protein synthesis is transcription. Then you need to know what happens during transcription, which is where DNA forms mRNA, and mRNA now has the coded message and for protein synthesis. So during transcription, we are just forming mRNA to copy the code from the DNA for protein synthesis. Then we have the second stage, which is translation. Translation is where the tRNA reads the code of mRNA and the amino acid links to form a protein. So translation is where now we are forming a protein using the amino acids, right? So that is what you need to know. Then the differences between DNA and RNA. When you take a look at this molecule here and this molecule here of RNA, the first one is of DNA, the second one is of RNA. You check what are the differences. This question appears to you as a tabulation. They may ask you to tabulate the differences between DNA and RNA. So here you must be able to draw your table and talk about the sugars. So when you speak about the sugars will be here. So you will say the type of sugar that is found in DNA is your deoxyribose sugar. Then the type of sugar that you find in your RNA is your ribose sugar. Then DNA is double helix. So you see here this side. So it is double helix. Remember, it's going to do this when it goes. So it is double helix in structure. Then the structure or the strands for RNA is single-stranded. So you must remember that. Then DNA is a very long molecule, whereas RNA is a short molecule. DNAs, the monomers, are nucleotides. Then what are the monomers? The building blocks. The building blocks are nucleotides, and then the monomers for RNA are also nucleotides. But with the nucleotides of DNA and the nucleotides of RNA, they differ. So now we need to check where do they differ. So then you can write that in your table. Right. So when you take a look at the nucleotides, this is the structure of a nucleotide, a basic structure of a nucleotide. Remember, when you go back, this is your entire nucleotide. Nucleotide is not just one molecule or one diagram. It comprises of three things. So let us take a look at what a nucleotide comprises of. The first group that a nucleotide comprises of is your phosphate group. So your phosphate group, then you have your pentose sugar. This is where now the difference between DNA and RNA comes. With your DNA, the type of sugar that you have is your deoxyribose sugar. But the type of sugar that you find in your RNA is your ribose sugar. So that is the difference that you can also put in your table when you differentiate between a DNA and an RNA. Then you find the nitrogenous bases. Now these are the nitrogenous bases that you can find. There are 4 of them, namely guanine, cytosine, adenine. Then when it comes to DNA and RNA, there are differences now. So with your DNA, you will find thymine instead of uracil. Uracil, you're going to find it in RNA. So this is another difference that you can also put in your table to say the type of nitrogenous base that you find in RNA is uracil instead of thymine. So in DNA, we find thymine instead of uracil. Please remember that. Now we get into the process. What is DNA replication? DNA replication, right. So here, the question will be for you to describe what is DNA replication. So you must give the steps. For about 5 marks or 6 marks, you must be able to collect the marks. The first 3 marks or the first 2 marks, I know it's easy marks for you, you get them. So let's go. The first thing that you need to say when you describe the process of DNA replication, you must say now, the double helix unwinds. So the double helix of the DNA, remember we said it's double helix in structure. And when you talk about the structure, please don't talk about anything else besides double helix or double stranded, or you're going to lose mark. Just say double helix. So the double helix unwinds. We see that happening here. Then the weak hydrogen bonds between the nitrogenous bonds are going to break, and then the DNA unzips. Here, you used to get 2 marks for saying each, but right now, for this entire sentence, is 1 mark. So for saying unzip, you're going to get 1 mark, or for saying the weak hydrogen bonds here between the nitrogenous bases break, you're going to get only 1 mark. And then, or you can just say the DNA molecule separates. Then, after the separation, each original strand now serves as a template. Now here you see the template being serving here. Now on which now a complementary strand is going to be built. Right. Now, how are we going to build the complementary strand? We're going to use the free floating nucleotides of DNA. Very important. So here you will have your, if this is inside your nucleus, you will have your free floating nucleotides of DNA floating. So those free floating nucleotides are the ones that are going to come and attach, form a complementary strand to the original strand. So the free floating nucleotides attach now to the original strands, now forming a complementary strand. Now we know how the bases pair. The nitrogenous base pair, A pairs to T, then C pairs to G. So this will happen as the free floating nitrogenous bases come You get a mark for that. Now, this results in what? In 2 identical DNA molecules. Remember, because the sequence of the nitrogenous bases was the same, so what will come here will form a strand that is similar to the other one. So DNA in this form, it has replicated. Now, each molecule now consists of one original strand and a new strand. So that is the essence of you explaining what is DNA replication. Then we come to another process that is very important that you need to be able to describe. Whether you can find a diagram on this one, you'll be able to interpret the diagram or be able to describe the 2 stages of protein synthesis. Firstly, let us take a look at our diagram. We have our diagram here. We know that this represents our nuclear membrane or double nuclear membrane. Then you have your DNA molecule in there, and then you have your mRNA. mRNA is labeled there. Then you have your codons. Okay, we'll speak to that when we explain. So let's just go straight to what happens during the first stage of protein synthesis. The first stage of protein synthesis is transcription. So transcription is the process where we are forming mRNA. That is why you are finding mRNA after finding your DNA molecule in our diagram or in our nucleus. Right, so what happens during this stage? Transcription is a stage that you can say it's familiar to DNA replication. However, there are differences. So you will see where the difference is or where you start to change when you explain the process. But for the first step, I say you should not lose these marks because you can get those 2 marks if you don't know the rest of the story. So transcription, double helix unwinds. The weak hydrogen bonds between the nitrogenous bases are going to break, and then the 2 strands are going to unzip or they're going to separate. So you get 1 mark for that entire story. So whether you talk about unzipping or whether you talk about the weak hydrogen bonds breaking, it's just 1 mark. Then 1 DNA strand is going to act as a template. Now, here is the first difference. Remember when we were talking about DNA replication, we were saying two strands are going to act as a template. But here, because we are forming 1 strand of mRNA, we say 1 original strand is going to act as a template to construct what? mRNA. How does it construct mRNA? This will be, sometimes it can be your compulsory mark, so you need to look for that to say the entire process is to form mRNA. So we want to see that in your entire explanation, right? How is it going to form that mRNA? It is going to use now the free floating RNA nucleotides. Remember with DNA replication, we were saying it is using the free DNA nucleotides. So the nucleotides in this case that are going to form the mRNA here are going to be our free floating RNA nucleotides. Then that mRNA is complementary to the DNA. What do we mean by that? So if the triplet base here was let's say we have your letters here, your triplet base. Remember, triplet base on your DNA are the 3 consecutive nitrogenous bases on a DNA. Then the ones that needs to come from mRNA are going to be the ones that complement the ones that you have from your DNA. Then the next stage, what is going to happen now with this mRNA that has been formed? And please remember that the 3 consecutive nitrogenous bases on your mRNA, we call them codons. And then the 3 consecutive nitrogenous bases on your DNA molecule, we call them DNA triplet base. Then this mRNA now is going to do what? Come into the next stage. What is the next stage? Translation. How do you remember this one? Which one comes first? This one you can just remember to say translation. Lation sounds like later. So you can say it comes after. So you will remember the first stage is transcription. The second one will be later, which is your translation, which happens in the ribosome. Then the first one happens in the nucleus. Right, what happens during translation? The coded mRNA, this one now, is going to leave the nucleus through the nucleopore here. It is leaving the nucleus through the nucleopore, and it is going to attach here on the ribosome. When it attaches on the ribosome, what is going to happen? tRNA, these ones now. tRNA is going to bring about the required amino acid. These ones, these black dots on top of a tRNA molecule are our amino acid. So tRNA is going to bring about the required amino acid by bringing the matching anticodon, the triplet based on DNA. Remember, we said they are anticodon. They bring the matching anticodon to the mRNA molecule. So if you have your mRNA molecule, this tRNA is going to bring about the required amino acid, then forming a protein. These amino acids, as they chain and chain and chain, they are bonded by a peptide bond, and then they form a chain of a protein. So this is how you form your protein. This is just for you to recap on the process, right? Then we come to DNA profiling. Remember, the term that we use now from your new exam guideline is DNA profiling. We no longer use DNA fingerprinting. Please, 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 please don't use that word. You're going to lose marks. Right. So what is this DNA profiling? This is a technique that is used to identify the sequence of the bases. Now, the nucleotides are separated from each other. Remember, the nucleotides are the building blocks, so we're going to separate them in the order that they are found in a strand. So in that order in which they are found, then those nucleotides are going to appear as dark bands. So if you see on our diagram here, you see those dark bands. These are your nucleotides. Then DNA profiling is then different from fingerprints, because now this carries your genetic material. Now, when you interpret this question, you need to have a ruler. You are going to get a question where you need to compare the profiles of persons or check which persons are closely related. The mother, you can check paternity. Remember the uses of DNA profiling. Then in order for you to get the correct answer, you put your ruler. Please make sure that you put your ruler. I know some of you would say your eyes are sharp, but please make sure that you put your ruler for accuracy. Then what are the key things that you need to remember? Remember, we are not touching everything. You need to go according to your exam guideline, but these are the things that you also need to remember for your revision. The history and the discovery of DNA, remember, by Watson and Crick, Franklin and Wilkins. So you must remember just that brief history of who discovered the shape, what happened with the X-rays. Remember that brief history. Then the structural difference between DNA and RNA, we've covered that. Then the differences between DNA, mRNA and tRNA are very important. So you need to be able to know those. Then you must also be able to differentiate between DNA replication, we have just covered that, and protein synthesis. Then know your stages of protein synthesis, transcription, translation. You must learn this off by heart. Please, these are standard in every exam paper. You are going to find these in your exam tomorrow. Definitely. Right. DNA profiling is very important. You may have questions, remember, where you have to use your ruler to analyze those DNA profiles. Then also know that paternity testing can be used from both parents to child, and then the uses and the interpretation of DNA profiling. So those are the things that you need to look out for. Look for questions in your question papers that cater for that. Then we come to the second topic, which is meiosis. Right. Meiosis, remember, in grade 10, you did mitosis, but now in grade 12, you are doing meiosis. You've done that, you know, you've discussed this with your teacher throughout the year. But what is this? This is the type of cell division where a diploid cell undergoes 2 cell divisions to form a haploid gamete. Very important. So you must not just say a haploid cell. We are forming gametes. So meiosis is the process where we are forming gametes. How do we form them? One diploid cell, 2N. Remember, it's 2N. It's going to undergo 2 divisions to form 4 genetically different haploid gametes. We are forming gametes. Right. Before meiosis begins, you must know that DNA replication takes place, which results in each chromosome consisting of 2 chromatids that are joined by a centromere. Now, you must know that this replication is the preparation stage for a cell division. Remember, we want our DNA to duplicate. We want it to double. So if you have a single-stranded chromosome, now it's going to be a double-stranded chromosome. So we will see how that happens. Then during the first meiotic division, what happens? One diploid cell gives rise to 2 haploid cells. So it's going to be one cell that is diploid, it is going to give rise to 2 haploid cells. Then you come to the second division. Then the 2 haploid cells, now these ones, the 2 haploid cells are now going to give rise to each 2 haploid cells. Then you end up having 4 genetically different haploid gametes. We call them gametes now at the end of this process. Then why is meiosis very important? This is a question. Every question of meiosis will end with this question, where they ask you, what are the importances of meiosis? Then we know. Crossing over introduces genetic variation, you will see. Then it also ensures that the sex cells have a haploid number of chromosomes. Remember, we need to have 23 chromosomes. So if meiosis does not happen, we will end up with organisms that do not, or cells that do not have haploid number of chromosomes. So meiosis is very important to halve that number of chromosomes from 46 to 23 and to keep it constant throughout the generations. So question 2 of your question paper, 1.2 is terminology. Give the correct biological term. So with meiosis, these are some of the terminologies that you need to be familiar with. Go back to your glossary at the back of your book and find out, what do we mean by homologous chromosomes or bivalent? Then chromosome, what is a chromosome? What is a chromatid? A centromere, a centrosome, crossing over, chiasma, karyotype, non-disjunction, karyokinesis, and cytokinesis. So we need to be able to know this terminology, and there's more that you can also study. Remember your crossword puzzles that you did throughout the year. This is the terminology that is going to come in handy. So we'll explain them when we go through the topic. Right. So during the interphase stage, remember we said this is a stage where preparation is taking place. So interphase is a step where the cell is going to do what? DNA is going to replicate, meaning we're going to make more copies. Then each chromosome will now consist of 2 chromatids joined together by a centromere. So we're going to have this stage. Interphase is very important to give us chromosomes like this, not a single-stranded chromosome, because now we want to double our genetic material. Now DNA replication helps to double the genetic material to be shared equally during cell division. We want to have enough so we can share. Then unreplicated chromosomes, single chromosomes, then are going to appear. Right, the story of unreplicated and replicated. By now you know. A chromosome, if it appears like this, it's a single-stranded chromosome. But if a chromosome appears like this, then you can only refer to this as a chromatid, and then this is a double-stranded chromosome. Right? So this is the story. This over here is your unreplicated chromosome. Remember the question that we always get at the end of the meiosis questions, where it asks you how many chromosomes are found at the end of this cell division. And then you find a cell with these types of chromosomes. And then you find people trying to connect this in their heads, putting a centromere that is not there and say 2.5 or 1.5. So we need to say these are chromosomes. We count them as 1, 1, 1. But they are now at an unreplicated stage, because from here, this cell is going to now undergo interphase again, where it's going to now double the genetic material again. So please take note of that. So when it is like this, when do you refer to a chromatid as a chromatid? Only when it is joined together by a centromere. Then you can say this one is a chromatid. But if it appears like this, it is not a chromatid. It is an unreplicated chromosome. End of the story. Right. So remember what happens during each phase. You just remember the differences. The very important stage is prophase 1. If you remember the events of prophase 1, you will know this plays a very important role when you have to answer the question of variation, because this is a source of variation. So what happens during prophase 1? The spindle starts to form. Remember, these are the spindle fibers, because you need them to pull your chromosomes at a later stage. And then chromosomes appear in homologous pairs. So you have the one from paternal, one from maternal, homologous, now you know. Then, or they are bivalent or bivalent. Then crossing over takes place. Crossing over, remember, is that process where the sister chromatids from homologous chromosomes touch at a point called the chiasma. Now, when they touch here and they overlap, they exchange genetic material. This process is very important as it brings about variation. So crossing over is a source of variation. You must remember that when you are asked about variation later. Then prophase 2. Not much is happening at prophase 2, but just remember now, this is the stage that happens at prophase 2. The nuclear membrane and the nucleus, they start to disappear or disintegrate, which happens also at prophase 1, but we were only focusing on the things that are very important there that you need to remember. Then here at prophase 2, there is no crossing over. Then metaphase 1, meta meaning middle. We'll say this is the middle. What you need to remember for metaphase is that the diagram for metaphase 1, you must have your chromosomes in that homologous pair, but now in the middle. That is the picture that you need to have. And then with your meiosis 2, you are going to have your chromosomes in the middle, but arranged singly. So how are they arranged? Now, the pairs of chromosomes aligned at the equator randomly. Now, this is also what you need to speak about when you are asked about variation. How does meiosis bring about variation? You will say crossing or the random arrangement of chromosomes that happens at metaphase 1. So, or at metaphase 2, because these chromosomes, they aligned themselves at the equator randomly. So please remember that. But the key thing to remember here, the differences between metaphase 1, metaphase 2, here chromosomes are in homologous pairs, but at the center, at the equator, here they are arranged singly, but at the equator. Moving on, anaphase 1, anaphase, apart. So you are expecting your chromosomes to be pulled apart, but let's see how are they pulled apart. The first one, here is a lot of story, so you need to remember what causes them to pull apart. The spindle fibers are going to contract. Going to get a mark for that. So the spindle fibers that are attached to the centromere or to the chromosomes are going to contract. So when they contract, the chromosomes are going to move now to the opposite poles. So you see this happening here. This one is moving there, that one is moving there, this one is moving there, this one is moving there. Remember, they were in homologous pairs. Now they are going as chromosomes, as a full chromosome to the opposite poles because of the contraction of the spindle fiber. Then two gametes, they are going, okay, right. This is the story that you need to know for non-disjunction. Remember, if the spindle fibers fail to separate, a pair of chromosomes, non-disjunction occurs. So if they fail to separate, non-disjunction is going to occur. What do we mean by that? Two gametes are going to receive an extra copy of the affected chromosome. That one that failed to separate is going to go into one cell and form an extra chromosome that is not needed there. And then if this happens at chromosome pair 21, it leads to Down syndrome. We call it trisomy 21. It leads to Down syndrome. Then at anaphase 2, it's the same story as anaphase 1, but now the chromosomes, remember from the previous stage, were aligned singly at the equator. So they will go singly. It will separate. Now the centromere is the one that is going to split. When it splits, it pulls one chromatid and one chromatid to the opposite poles. So you see that happening here. It doesn't go as a chromosome because this is now at anaphase 2. Then telophase 2. Telophase 2 is where now the nuclear membrane reforms. Where is it going to reform? Around those chromosomes that were pulled to the opposite poles. So it's going to reform, and then the cell membrane is going now to constrict. So the cell membrane, remember, is this part that is on the outside. So that cell membrane, when it constricts, it forms what we call a cleavage. Now you can already see what is going to happen here. So cytokinesis, okay, cytokinesis is the splitting of the cytoplasm. Then you end up with two cells at the end of the stage, meiosis 1, telophase 1. Then you bring those back to telophase 2 from those stages. So at telophase 2, remember we had those from anaphase, they were single, separated there as chromatids. So what is going to happen? Each daughter cell has half the number of chromosomes. Remember, we are halving. Meiosis is a halving process, so we're halving the number of chromosomes. Then we're going to form the cleavage still And then when you form a cleavage, remember the cytokinesis also occurs. It splits the cytoplasm. You have 2 cells. You have 4 cells at the end. But these 4 cells are all going to have half the number of the initial chromosomes that you found in a cell. And remember, when they ask you how many chromosomes here, you are going to say 2 if this is one cell. You are going to say 2, not 1. You're not going to combine these 2 in your head and say they form 1 chromosome. And when you say 2, you can check. It is actually half of where our cell is coming from. It initially had 4, but now you have 2 at the end of each cell. Now, these 4 genetically different or 4 haploid daughter cells are genetically different. So you can see even after crossing over that they are not genetically different. So this is how you get your gametes. Now at this stage, now we call them gametes. So what is non-disjunction? We spoke about non-disjunction, which is a failure to separate of chromosomes. So the spindle fibers fail to separate during meiosis. That is your non-disjunction. Now, members of one pair of homologous chromosomes, they fail to become separated during anaphase. Remember, anaphase apart. Right. So when that happens during anaphase 1, or it happens also in anaphase 2. Remember, in anaphase 2, it will be single chromosome. Anaphase 1, homologous chromosomes. Right. If there is non-disjunction of chromosome pair 21, now very important this one. When there's a failure to separate at chromosome 21, in humans, it leads to the formation of abnormal gamete. Why is the gamete abnormal? Because we say this gamete is going to have an extra copy. Remember, a gamete, we want it to have 23 chromosomes. But our gamete, if it has an extra copy, it may have 24 chromosomes. And then what is going to happen if this gamete fertilizes a normal gamete? This is where now we're going to say it leads to Down syndrome. But we know some of you like to speak of this in the negative. Please don't. When you focus on chromosome number 21 for Down syndrome, focus on the additional, on the extra chromosome. Because if you say there is less and then you have a cell with 22N, you are no longer talking about Down syndrome. You are talking about another genetic disorder. So please, for my meiosis or at chromosome 21 for Down syndrome, please talk about an extra chromosome or a gamete having an extra chromosome and then fertilizing a normal gamete, then leading to Down syndrome. Now, 2 gametes are going to receive extra copy that are affected by the chromosomes, and then 2 may lack that chromosome. So this part of lacking, this is just for you to understand, but it does not necessarily explain Down syndrome. So for Down syndrome, we need that extra one. Please focus on that. Then what happens during non-disjunction? This is just a diagram to show you. During meiosis 1, we have 2 chromosomes under normal separation or under normal circumstances here. This was supposed to happen. You see here, there were supposed to be 1, 2 separated chromosomes. But with non-disjunction, you'll find that they failed to separate and they both came into one cell and then they left the other cell with nothing. So this is non-disjunction with chromosomes. Remember anaphase 1. Then with anaphase 2, which happens in meiosis or meiosis 2, you are going to find that these chromosomes here, they were supposed to separate, one in each cell. But look at what happened. They both came into one cell. And remember, at the end of meiosis 2, we are now talking about gametes. So this is where you're going to find a gamete with an extra copy, then another gamete with nothing. So this is just a diagram to show you under normal circumstances, we would expect these gametes to separate, and then each cell will have 1 chromosome as illustrated there. Right. So what are the differences between mitosis and meiosis? Remember, it's a key thing that we said we need to look upon. Now, firstly, mitosis occurs in the body cells. Right, somatic cells. Then meiosis occurs in the sex organs. So your ovaries and your testes. So the organs, not the sex cells. Please make sure that you know the sex organs. This is where you'll be talking about your ovaries and your testes. Then here are your ordinary body cells, which we call them somatic cells. This is mitosis. Then mitosis also, it has both karyokinesis and cytokinesis. It occurs, they both occur once. So karyokinesis is the splitting of the nucleus. Then cytokinesis is the splitting of the cytoplasm. Then also in meiosis, karyokinesis and cytokinesis occur, but they occur now how many times? Twice. Here they occur once. Then 2 daughter cells are formed at the end. 4 daughter cells are formed at the end of meiosis, which we call them gametes. Now, the daughter cells that are formed that are 2 in mitosis, they are genetically identical. They are identical even to the parent cell. However, in meiosis, the 4 daughter cells that are formed are genetically different. Each one of them is different because of crossing over that took place at prophase 1 and also random arrangement of chromosome at metaphase 1 and 2. Right. Chromosome number remains constant. So if it's 4, 4, 4, 4, they will produce 4, 4. But here the chromosome number is halved. So if you started with 46 chromosomes, you must end up with 23 chromosomes at the end of meiosis. Right. Those are the key things that we needed to study for mitosis and meiosis. Then this brings me to the end of my presentation, but we are going to continue with the rest of the topics. Mrs. Vedika Maharaj is going to take you through the rest of the topics that you need to cover for your paper 2. Thank you.
Welcome back, grade 12 learners. My name is Vedika Maharaj, and I'm going to take you through the next 2 topics that you need to study for your paper 2 on Monday. Right, so let's start with the genetics and the inheritance. It's 48 marks, as Sina has told you earlier on in the session, which means it's basically a third of your paper. Okay, it's a large chunk of your paper, so it's very important that you understand this topic and that you study it well. So here's a little mind map of the subtopics that you need to know. Remember that this is just an overview. You must look at your examination guideline, which will give you an indication of how much detail to go into each subtopic. Okay, so just to go quickly through them, here we are looking at terminology. Important because many learners confuse some important terms. We have monohybrid crosses, sex determination, sex-linked inheritance, dihybrid crosses, blood group, genetic lineages and pedigree diagrams, genetic engineering, paternity testing, genetic links, and Mendel's principles of inheritance. Not in this particular order, but just to show you these topics, please make sure that you know them. They should ring a bell as I've mentioned them. Right, this just gives you also a bit of an overview on your genetics and the topics that you need to know. Right, moving on to the terminology. These are not the only terms, but these are the terms that are commonly confused. Learners commonly, they confuse them, they use them interchangeably. Make sure that you know the difference between chromatin and chromosomes, genes and alleles, dominant allele, recessive allele, phenotype and genotype, and homozygous and heterozygous. Okay, we have gone through this before. Your teachers have taught you these terms. We're just showing you that these ones are used interchangeably and are often confused. Right, this is a question that is most frequently asked. It's Mendel's laws of inheritance. Now, you remember the man over here, it's Gregor Mendel, and he actually did some observations and some experiments on pea plants, and he basically came up with these 3 laws. You need to know them. You will find them in your examination guideline, and you need to be able to know them word for word from there. So if you are asked to state Mendel's first law of inheritance, which is the law of segregation, then you need to give it word for word from the examination guideline. The same with number 2, Mendel's second law of inheritance, which is the law of dominance, and number 3, Mendel's third law of inheritance, the law of independent assortment. Make sure you know them, and make sure you also understand them, so that if you are asked to explain it in your own words, then you know how to do that as well. Right, now we're looking at the format of a genetic cross. Okay, it's like a 90% assurance that you're going to get a monohybrid cross that you're going to have to work out. Okay, so they can ask you it on different things, different types of inheritance, which we are going to look at. But I just want to start with the format to remind you the important things that you need to remember when you are doing your genetic cross. It's usually about 6 marks. 6 or 7 marks, and if you look at it as that amount, as 6 or 7 marks, ask yourself how many. I'm telling you now that going into your paper, no matter what question you're going to get on monohybrid cross, if they ask you to draw a genetic cross, you are guaranteed 3 out of that 6 or 7 marks. Where are those 3 guaranteed marks? Right, so we're first looking over here at P1 and F1. Okay, you get a mark for that. Then you get a mark for meiosis and fertilization. What's important is that you must remember that the P1 comes before, it's at the top of your genetic cross, because it's referring to the parent generation. Whereas the F1 is the offspring or the filial generation, so therefore it is towards the bottom after fertilization has taken place. The next thing to remember is the place where meiosis and fertilization fit. Meiosis happens before fertilization. Okay, so those are already 2 marks, right? I call them Mahala marks, because these 2 marks you go into the paper. It's nothing that you study for. Know the template, okay? Know the template, know the recipe. Then let's look at our first line, phenotype. What is the phenotype? The phenotype is the visible trait. Okay, it's the visible trait, for example, tallness or shortness. This you will get from the question, right? So there we have another Mahala mark. You see, 3 marks. You're already going to the paper having those 3 marks for this question. Then thereafter, it's just a matter of working out your genotypes, which is the genetic makeup. And thereafter, we have our law of segregation that's going to come into play here, where the alleles are going to separate during meiosis to form our gametes. Right? And then during fertilization, the individual gets 1 allele from each parent. So during this process of fertilization over here, we have the genotypes and the phenotype. So we have these individuals that are going to come out from this fertilization. They will get an allele from each of the parents, which is coming from the genotype essentially. Okay, so this is one version of the cross that you can do. And if you don't like that one, you can also replace this fertilization part with this Punnett square. Okay, if you do, what you do over here is you plug your gametes in. These gametes, you plug them into here and here. This is where the gametes are plugged in, and then you cross them and you get these individuals. Right, those are the difference. So I also want to stop here and just tell you that your question paper is, if you look at your question paper and you say it's 150 marks, it's like this mammoth task that is ahead of you. It's a great task. It's like a giant that you need to overcome. But if you look at your question paper in smaller sections and in smaller parts, for example, 2.1, let's look at 2.1. From the whole of 2.1 is going to count maybe 15 marks. Ask yourself, and you must be confident that from that 15 marks, you can definitely get at least half of those correct. Our questions in life sciences are scaffolded. So you are going to start with your easier questions at the top, and then we move within the question. We have more easier questions. We start with labeling or something that's a recall question, and then we move down to a little bit more difficult things. So if you look at your paper in smaller sections and you tell yourself that you can overcome these smaller sections, then it's not such a giant that lies before you. So that's just to remind you about where the marks are, and then we move to the types of inheritance. Okay, so we spoke about the monohybrid crosses. Now within that, the types of inheritance we get there, we are looking at these three, which are complete dominance, incomplete dominance, and co-dominance. Okay, oh yeah, and we also have sex-linked. Now you can get asked to do a monohybrid cross, and you can, that's going to be on any of these. Okay, I'll say that again. You'll be able to do that monohybrid cross, that format that I just showed you. You can apply that skill of that format to any of these questions that they can ask you. It doesn't necessarily mean that they're going to give you one on complete dominance. They could give you one on incomplete dominance or co-dominance, or they can give you a sex-linked one. Okay, so we're going to go through them quickly. You've done them already, right? So we're just going to go through them quickly. Oh, before we do that, I want to also indicate to you that there are specific wordings to use when you need to explain complete dominance, incomplete, and co-dominance, and you get them in your exam guideline. Please have a look at those definitions and make sure that you know them. Right, the complete dominance. Essentially, what complete dominance is telling us is that when we have a green pod, a true breeding green pod, so we have two dominant alleles here. Okay, we have two dominant alleles here, and they are indicated with a capital letter. Right, we have an individual that has two dominant alleles. It is homozygous. Okay, and it is crossed with an individual that has two recessive alleles, small letter. Okay, I'm just going to put it in here, recessive, and then you have your capital, which is your dominant. So when we have the homozygous dominant crossed with the homozygous individual, all the offspring will be heterozygous. Okay, and also, more importantly, they will have only one parent's phenotype will be evident in this offspring. Okay, so the one that is evident, that is the one, in this case, it will be the dominant allele, because it's heterozygous. So we have one dominant allele, one recessive allele. So therefore, the dominant one is going to mask the recessive one, and all the individuals are then going to have the dominant trait. Okay, that's the law of complete dominance. If you were asked to do a monohybrid cross, you would do the same thing as the format that I've shown you before. Okay, you plug in all the necessary information, and then you work out how they would be. You do not only do a Punnett square, right? You do the whole recipe. Right, now we're looking at incomplete dominance. Now here you'll see something funny is going on, right? Previously we had green and we had yellow, and then everybody was at least one color, right? Which was green, one color of the parent. Whereas in this one, we have a red flower and we have a white flower. And what they found, that if you cross these particular ones, all the flowers of the offspring are pink, right? Which is a blended phenotype, because if you blend the colors red and white, you're going to get pink. Okay, so it's a blended phenotype. Another important thing for us to remember here is the letters that are being used. When it comes to incomplete dominance, we are using different letters for different color trait. Okay, or for different traits. So in this instance, the red is capital R. Okay, and then the white is capital W, and we only use W's. And can you see we're using two different letters? Whereas in complete dominance, we used one letter and we differentiated between dominant and recessive. Okay, here we don't. Why? Because in this instance, we have neither of the parents' phenotype that is evident in the offspring. Okay, it's not that we see red and we don't see white. We see a blended phenotype. And this indicates to us that neither of these alleles are dominant. Okay, neither are dominant. Therefore, we have a blended phenotype. Then we move on to codominance, right? When we have codominance, in this instance, we are also using letters, okay? We are using different letters for the different traits. But here we have the offspring, both the parents' phenotypes are evident in the offspring, okay? So if we have a white cow and we have a brown cow in red one, sorry, in this case, we get a roan cow in the end. So we have this white cow with these red spots. So you can see white there and you can see red there, right? And this means that both of these alleles are equally dominant. Okay, so just to show you quickly, the way I remember it is, if we are looking at complete dominance, okay, if we are saying that this trait, which is the dominant one, and this is the recessive trait, the dominant trait always masks the recessive trait, okay? When we come to incomplete dominance, we have a blended phenotype, okay? It's blended. We have two traits, they are blended. When we have codominance, they are equally dominant. Therefore, they are equal, right? They are equally dominant, they will both be. You can see that we will now see both of them in the phenotype. Okay, so like I said, we can ask you any monohybrid cross to work these out. Just remember those little tips that I gave you. Right, now we're looking at sex determination. So this is called a karyotype, right? It's a representation of all the chromosomes that are found in a human cell. And the important thing here that I want to mention to you before we actually get to the sex determination is that, remember, in a cell, in a diploid cell, we have 46 chromosomes, right? Which is basically 23 pairs. Right, now what I want to show you is that 44 of those 46, right, are gonosomes. Autosomes. Sorry about that. Are autosomes. 44 are autosomes. And the two that is left over from this 46, those are the gonosomes. Okay, so in this particular karyotype, they are showing you all 23 pairs, all 46 chromosomes. So all of these here, all the way until here, these are the autosomes. This one, these are the gonosomes, and these ones determine whether the individual is going to be male or female. So, how do we know the difference? When you look at those two gonosomes, you will see that if it has an X and a Y, then it will be a male, right? And then the next picture that I have for you is an X and an X. You can see over here that's indicated in pink. The XX is indicating a female. Okay, so it's important to understand that you can use it. You can see it happen in meiosis, where they ask you to calculate how many. They give you maybe a scenario of a diploid organism, of a diploid cell of a chicken, for example, that has maybe 20 chromosomes, and then they ask you other questions on it. You must always understand that we have, like in a human, for example, we have 23 pairs, which are 46 chromosomes. 22 pairs or 44 chromosomes are your autosomes. And then the other two, the last pair, are your gonosomes. Okay, so that is a sex determination. And also further, the important thing about sex determination is that you need to know there is a 50% chance of having a boy and a 50% chance of having a girl. Okay, and you can be asked to prove that. You can actually be asked to prove the statement. Okay, and when you're asked to prove it, you use a genetic cross or you use a Punnett square to actually prove this. Okay, that you have a male with XY and you have a female with XX, and then when you cross that, you will see that you'll get two girls. So it's 50% girls and 50% boys. Chance of having boys or girls. Right, sex-linked disorders, very important. So you will see that sometimes you when you're doing your genetic crosses and you have a male and you have a female, right? And then you've got like superscripts here, right? Looking like that. Okay, so these are showing you the genotypes. Okay, these are the genotypes, and then these are the alleles, right? Okay, so we use these notations when we're using sex-linked disorders. Okay, so just to remind you what they are, these genes are located on sex chromosomes and they are called sex-linked genes. Okay, they are not found on other chromosomes. They are only on sex chromosomes. In humans, the term refers to X-linked characteristics, and the fathers can pass down the X-linked alleles to their daughters, but not their sons. Why? Remember the law of segregation. It says that the offspring is going to get one allele from the mother and one allele from the father. So if this particular offspring is a boy, right? Remember, his genotype must be X. His chromosomes must be XY, right? Where does he get that Y from? He can only get that Y from his father, right? Because the mothers females have XX. So that boy could only have gotten the Y chromosome from the father, which means that his X chromosome has to come from the mother. And if this particular boy got the recessive allele that is maybe carrying a disease, then if he gets the one from the mother that is carrying that recessive disease allele that is causing a disease, then this individual will have the disease. Okay, so that's what they mean by fathers can pass the X-linked alleles to their daughters but not their sons, because this X of the male, this one can go to a daughter because it must get one from the father and it must get one from the mother. Either this one is coming from the father and any one of these will come from the mother, either the H or the small h, capital H or the small h, right? And the mothers can pass the sex-linked alleles to both their sons and their daughters. Why? Because like we said, they must get one allele from the mother, one from the father. So she can either pass this one or that one to either the son or the daughter. Okay, so this is the two sex-linked disorders that you need to know that is part of the examination guideline is hemophilia and color blindness. So if you are asked a genetic cross and they give you a scenario link and they tell you that it's hemophilia or they tell you it's sex-linked, you must remember that you need to use your X and Y and your X and X. Your X and Y as alleles for your father and the X and X for the mother. Not all genetic crosses are sex-linked. Okay, it's not all. They will indicate to you, unless it is hemophilia and color blindness, then you need to know that it's sex-linked. But if it's another question and it's not one of these two, they will give you an indication in the question that they are looking at sex-linked. Okay, so and that's just another picture to illustrate to you what I explained on the previous slide. Okay, so this one is for color blindness, just to remind you once again that these two are the sex-linked diseases that you need to know. Right, now we are looking at blood groups. Now, the inheritance of blood groups is an example of multiple alleles. Okay, why? Because in this instance, we have alleles that the notation for the alleles indicating blood groups is only these three. Okay, so we have these two are dominant. Okay, and this one is recessive. But can you see how many there are here? There are 3 different alleles. Usually we only have 2 different alleles. So therefore this is multiple alleles, right? Because we have more than 2. So the different combinations of the alleles result in 4 blood groups. So if you have to combine these in this way over here, you'll look at it over here, we'll look at it just now, but you combine them in different ways, you can get a possibility of 4 different blood groups. So the blood group is the phenotype. Okay, so if somebody is blood type A, that is their phenotype. Okay, or blood type B, or blood type AB, or blood type O. These are not alleles. So learners do that. Okay, they write, when we ask blood type A, then they start writing the A's and the B's. Okay, those are not the notations we use for the genotype. These are the notations that are used for the genotype. Okay, so with blood type A, with blood type A over here, we can have two options, okay? Because remember, we either have a homozygous condition or we have a heterozygous condition. Homozygous meaning that the alleles are the same. So in this instance, these two are the same. It's both A, so therefore it is blood type A. This one is showing one dominant and one recessive, okay? And the recessive, one dominant and one recessive. So the dominant is going to mask the recessive, and will be blood type A. Then we have blood type B is a similar thing to blood type A. We have AB. Here in AB, what's important to notice here is that they are different, okay? But they are both the dominant ones. And then lastly, we have homozygous recessive, where we have two recessive alleles, and that is your blood type O, okay? So the important thing I want to bring to your attention here is that when we are looking at the allele, the one for this allele here and that allele, this one is showing us complete dominance because the dominant allele is completely masking the recessive allele, okay? The same with that one and the same with O. So A, B, and O, we have this complete dominance. But with A, B, we have co-dominance. These are equally dominant. Why do I say that? Because they are both dominant and they are both expressed equally in the phenotype. That's why we have these two notations, and it comes to blood type A, B. This is also a genetic cross that they can ask you to do. They can also give you a monohybrid cross on this. Okay, so they'll tell you what the what the parents are, and then you will work out the cross. Right, genetic lineages and pedigrees. Okay, a pedigree, it traces the inheritance of characteristics over many generations. Okay, so it's basically like a little family tree. Now, when learners see this, okay, let me first explain to you what is happening here. What's happening here is that it's indicating to you different symbols. Okay, pedigree diagram also. A very good chance that you're going to interpret this kind of a question. So what we're looking, I'm going to put the next picture to show you what we're actually looking at here. When we are looking at a pedigree diagram, what it's indicating to you is, firstly, we need to know that the circles are females, the squares are males, and when it is shaded, then they are affected with that trait. Okay? So they can also ask you to do a monohybrid cross based on this information. They'll tell you that this person X, person X is, no, let's not use X. Let's say person B is marrying person M. Okay, and then they're going to have they're going to have offspring, and they're going to tell you what characteristics they have, and you need to do a monohybrid cross. Okay, but the important thing to understand here is that it's not always sex linked. Okay, even though it's a monohybrid cross, and because they're showing you a male and a female, I don't know, for some reason learners think that this is now going to be sex linked. Okay, not necessarily. Remember what I told you, they will indicate to you. Right, then this line over here is indicating a marriage line. This one is the line of descent, and then here we have a sibling line. So these two are the offspring of these two, and these two are siblings. Okay, and then here we show indicating to us the generation number. So when they like to ask that question, let's go to the previous one quickly. They like to ask that question, how many generations can we see here? So we count them as lines. This is generation 1, this is generation 2, and this is generation 3. Do you see that? Okay, so those are the important things that we need to indicate to you. They don't always give you this, therefore you need to know what these symbols stand for. Okay, these ones we don't really use very much. Those are the main ones that I explained to you. Okay, we're not going to spend a lot of time on this, but it's just the steps when solving a pedigree diagram. The main important things to remember is that we study the key. Okay, we look for which are the dominant and the recessive characteristics, and the easier one is to first identify the recessive ones, because the affected ones, because usually if that is the recessive allele and they are affected by the recessive allele, then if they are homozygous recessive, then those will be the affected ones, the ones that are shaded in, and then you just work it back. Okay, so that you can actually work out what the genotypes are going to be. These are the kind of questions that they're going to ask you. Okay, so if the parent has the dominant characteristic, you fill in a capital letter, okay, and then the small letter will be the recessive characteristic. Right. Then we go to paternity testing. Okay, now this is the question that they also like to ask you. Okay, it's paternity testing, and the important thing that you need to understand this is that they actually use two. Okay, they either use blood groups or they use the DNA profile. Okay, but it's important for you to understand that with blood groups, it's not a definite answer that you will be able to indicate who the father is by identifying the blood group. Because there's many people that could have the same blood group as those four men that are maybe saying that they are the father of the child. Okay, but what it can do is, if there are four people that are thinking that they could be the father, okay, it can indicate maybe which one is not the father. Because if we work out a genetic cross and we say, okay, fine, the mother is blood type O and the father is blood type AB, right? But the child is blood, and the possible father is blood type AB. The child is then maybe blood type O. Then that father cannot be the child, the father. Let me quickly explain it to you. The mother is blood type, I don't know if you can see there. Let me do it here. The mother is blood type O. Okay, and the possible father, this I'm going to put an X here, a question mark to show you the possible father, is blood type AB. Okay, so the offspring, we know that the offspring is blood type O. Now, this one, it had to get one allele from the mother. So yes, it got this allele from the mother. But the other allele must come from the father. And this particular father does not have that allele. So therefore, he cannot be the father. So I'm just showing you that is how you can actually, they can ask you an application question like that, and that's how you will go about it. When it comes to DNA profiling, what you need to do here and how you need to explain it is that they've got the mother's DNA, they've got the child's DNA, and they've got the possible fathers. Okay? The first thing you do is identify all of the bands that match the mother. All the child's bands that match the mother. So in this instance, we have this one, we have this one, and we have this one, and this one. All of those match all of those bands of the child match the mother. The remaining bars of the child must match the father. Okay, so let's look at this one. We have this one matched with father 2. This one also matches with father 2. This one with father 2. That one. Okay, these ones, okay, that one doesn't, and then these two. So can you see? It's definitely father 2 that is the father, because so what you do is you need to understand that, because they can ask you to do it to say it in words. That you are first going to, the child's bands, you first check which ones match the mother, and the remaining bands must match one of the fathers. Okay, and the ones that match the most or all will be, that father will be the father of the child. Okay, so that's paternity testing. Right, now we move on to evolution, another large topic, 54 marks, okay? But you know what? There's some nice questions in here. Oh, they're all nice questions, but there are some, you'll see there's some tips here that I'm going to give you, and important things that you need to remember. Right, so first we start with our evidence of evolution. Okay, you should all know this. We have biogeography. You studied all of these: biogeography, genetic evidence, homologous structures, and the fossil record. Okay, I just want to remind you, and we'll look at it later on, that biogeography is a line of evidence that also can play a role in speciation. Okay? Then we're moving on to sources of variation. So Sina started with this because she actually told you the two meiosis ones, which are crossing over and random arrangement. Okay? But I put it in this table form for you just to remind you that when we are asked a question on sources of variation, okay, a nice acronym for you to remember is CRRRM, okay? And then the first C starts for crossing over. The first R stands for random arrangement, and these two take place in meiosis, okay? Meiosis. Then the ones that are related to the source of variation related to reproduction are random fertilization and random mating. Okay? And as you can see in the pictures actually explain to you a little bit more. Crossing over and random arrangement, Sina has done in detail, so I'm not going to go through it. Here in random fertilization, you can see there's one ovum and there's millions of sperm that are swimming towards it. Okay, so which one, it's not like they are numbered, which one is going to go first. It's random. Whichever one gets there first and can penetrate the ovum will successfully will enter. And random mating as well, even though these are all the same species, okay, it's not like it's random, whichever one that you can be chosen. Okay, and then lastly we have mutations. Okay, and important just to mention to you, a genetic mutation, it's important to know that definition. Okay, it is the change in the sequence of the nitrogenous bases of a DNA molecule. Okay, that is how you must say it, and that is the correct answer, right, for the genetic mutation. So yeah, that's for sources of variation. Then we come to different types of variation. We have two different types. We have continuous and discontinuous. Now, they like to ask this question as a table. Okay, sometimes they like to ask you to tabulate the differences. Please remember that when we have a table, you are going to take a ruler and you're going to draw a table like this. You get 1 mark for just doing that. Okay, the next thing you need to do is you need to put your headings in here. Okay, and when you compare things, you need to compare the same things. Okay, you can't talk about something here and talk about something different here. I'll do it in more detail when we go to another topic just now. I'll remember to mention it to you there. So continuous variation, these are where characteristics are in a range of intermediate phenotypes. For example, a nice one to look at is height. Okay, it's not that if you are tall, you are 1.6 or 1.9. Or if you're short, you are 1.5. Okay, there's a range of different phenotypes. Okay, that is going to be for height. And with discontinuous variation, this is when we have distinct specific categories. For example, you are either blood type A, blood type B, blood type AB, or blood type O. Okay, there are specific characteristics. There's no range. Right, now we move on to the theories of evolution, starting with Lamarck. Okay, so we have Jean-Baptiste Lamarck. Okay, you all know him. Please know his 2 laws. Okay, exam guideline, word for word. The law of use and disuse, the law of the inheritance of acquired characteristics. Furthermore, you need to know how to answer questions based on his theory. First, you need to understand what does inherited characteristic mean and what does an acquired characteristic mean. An inherited characteristic is the one that the offspring is born with. So it's a characteristic that you are born with. But an acquired characteristic is one that the offspring is not born with and you acquire it in your lifetime. So for example, if you are you're not born with if you are a bodybuilder, you're not born with those big muscles. You work out and you train, okay? And then you acquire those muscles. So that's the difference. And then the guiding questions. So you ask yourself, firstly, when you answer this question, what was the original characteristic? Okay. So basically, let's look at the giraffes, for example, when we answer this question, okay? We have Lamarck that said that the original characteristic was that all giraffes had short necks. And the challenge was, then what is the challenge? The challenge was that the trees, they had lots of leaves high up in the trees, and the organism, what did they do to acquire the characteristic? They were kept on stretching their necks to get to the leaves at the top, and that caused them to have elongated necks. So that's an acquired characteristic. Okay, so what happened to this acquired characteristic? They passed it down to their offspring. Okay, that is what Lamarck is basically saying. But you need to know that it was rejected. Okay, that his law was rejected, and you need to know why. The main reason, number one, that there was no evidence that acquired characteristics are inherited by offspring. And basically, they do not evolve because they were determined to change, but changes take place due to random mutations. This is a very important thing that we need to understand about evolution that I think learners don't understand, is that these mutations and these changes that occur are because of random mutations. Right. Then we come to, so that was Lamarck, okay? He had those 2 laws. You need to know how to apply it, and then you need to know that they were rejected and why. Now we come to Charles Darwin, okay? And he has his theory of natural selection. Okay, so you hear natural selection, you think Charles Darwin. You hear Charles Darwin, you think natural selection. Right. Basically, he says that species were not created in their present form, but evolved from ancestral species. Okay, like for example, the whale. This is what the whale originally looked like. Okay, and over time, because of random mutations, because of environmental factors, they changed. And number 2, he proposed a mechanism of evolution which was called natural selection. Okay, now, when you get a question on natural selection and you get asked to describe the process of natural selection, you take it straight from the exam guideline. Unless they give you a scenario like in this question. They give you a scenario and they ask you to use Darwin's theory of natural selection to explain the evolution of the lizards with larger jaws. Okay, you need to take that recipe from exam guideline and you need to apply it to this particular question. Okay, so you need to know the key words for the recipe. Firstly, you mentioned that there's variation. Okay, you state where the variation is. In this particular case, it's the lizard's jaws. Then you state those 2 different characteristics: small jaws, large jaws, right? Then you talk about what happened, right? There was a large supply of fibrous plants, or you can say fewer insects. Those with smaller jaws will be unable to feed and they die. The lizards with the larger jaws will have more food and survive. To reproduce, the allele for larger jaws will be passed on to the offspring. The next generation will have a higher proportion of lizards with larger jaws. Okay, the important thing I wanted to show you in this one is that everything, except for the things that are basically highlighted, all of that is basically the generic answer from your exam guideline. So learn that. Learn those different points and apply it to whichever question they're going to give you. Make sure that every sentence you have is specific to the question that was given. Right, then we have equilibrium, punctuated equilibrium. It's another theory that was developed. And in this particular one, so Darwinism is basically gradualism. Let's do that one first because we were talking about him. Basically, what he's saying in gradualism is that we have this mouse here, it's a beigey color one or brownie color one, and then we have changes that occur gradually over time. You can see that phenotypically they are changing to either become the white one or they branch off, they developed another mutation, and then they developed into that darker one. But can you see that the change is gradual? Whereas in punctuated equilibrium, they are saying that we have periods of rapid change where things change drastically, and then we have long periods where there is very little or no change. So we can see that in that the previous picture with these mice is that we have this brown color mouse, and then we had this rapid change. Can you see from this brownish beige color we went to white, or further up we had another change where we went to this brown one. So this one is showing us these are the rapid change and these ones are the equilibrium. It's not always going to look like this. Okay, you need to analyze your diagram in order to see how which one they are. Then we have artificial selection. Okay, in this one, it's important for you to know here is that you need to know artificial selection in domesticated animal species. A good example for you to explain is the one about the gray wolf. The ancestor of all domesticated dogs now is the gray wolf. Okay, and then humans were physically breeding them for specific characteristics, and that's how we landed up now with all these different breeds of dogs with these special characteristics. And then you need to know one example of a crop species, and this one that we like to use is the one about the mustard plant, and then humans physically breeded the plants with specific characteristics to give rise to these vegetables that we have here today. Right, moving to speciation. Learners commonly confuse when to use the word species, when to use the word population. Okay, so this is in the question on speciation, which leads to this point over here. Okay, but let's first understand what's the difference between a species and a population. So here we look at, let's look at this diagram to explain it. Okay, so this organism is of a particular species. Okay, other organisms that have the same characteristic as him, that are capable of randomly interbreeding and to produce fertile offspring, that is a species. Okay, so the keywords there are same characteristics, capable of interbreeding and producing fertile offspring. Whereas a population, this one here shows us a population. These are the individuals of the same species and they're living in a particular area and they can still randomly interbreed. Okay, so it's the same species living in the same area. That's a population. Now, speciation is an evolutionary process by which the populations evolve to become distinct species. Okay, so this is a process after many, many years that the population of this particular species, because that population is exposed to different conditions, they are going to change over millions of years and over a long period of time to eventually become different species. And how does it do that? It does that by geographical isolation and reproductive isolation mechanisms. These isolate the gene pool of a species, resulting with the formation of a new species. Okay, now with that information, let's bring it quickly into speciation, right? We've already done this. We've done this in class. I just want to remind you the important things and the place where we use the wrong words. Okay, so when we are looking at speciation through geographic isolation, once again, this is the recipe from the examination guideline. Okay, if you get asked to describe speciation, you're going to give the generic answer, but 95% of the time they're going to give you a scenario and they're going to ask you questions on it. Okay, so you're going to give them, this is an example. The important thing here is to say that there was a population. The population is split into 2 or maybe 3 or 4, whatever the question is going to give you. Okay, the population is split by a geographical barrier, not a species. You don't say the species was split, you say the population. You will lose marks if you're going to say a species here. A population is split into 2 by the geographic barrier. There is no gene flow between the 2 populations. Exposed to different environmental conditions, each population will then undergo natural selection independently. Let's look at this diagram to explain this. This is the ancestral population. This is the geographical barrier. Be specific to what the geographical barrier is here. The population is now split. There are 2 groups. 1 group, 2 groups. They are going to have different environmental conditions. They are going to go through natural selection independently. And then they are going to change genotypically and phenotypically. And even if they were to mix again, they are unable to interbreed, and then there will now be different species. Okay, we're not going to go through this question. I just put it here to remind me and to remind you that biogeography plays a role, okay, it's a line of evidence that can play a role in speciation, right? So in this instance, and what are we saying here basically? We're saying that if these 3, coyote, jackal, and dingo, basically what the question is saying is that they are now on different continents, right? But they share similar characteristics, which leads us to believe that they were once on a common, they once shared a common ancestor, and they were once on a large landmass. Okay, and then they got separated and they changed. So you need to know how to apply speciation to how to apply biogeography to speciation. How do we do that? Okay, just want to show you what was different from what we previously did in specifically. Firstly, we are looking at the type of evidence over here is biogeography. And when you have to describe speciation, you need to say that the original population once lived on a large continent. Okay, this is the part, these 2 points are linking to the biogeography. Okay, and then they became separated by the ocean or by the continental drift. Okay, another thing I want to just bring to your attention here is that always be specific, because in this one they wanted you to say that they were split into 3 populations, not just, the generic one you always get is that it was split into 2, but it was split into 2 populations, and there was actually a compulsory mark for this. And then at the end, always say what did the species actually develop into, because that is our other compulsory mark. Right, mechanisms of reproductive isolation, usually asked in section A. Please make sure that you know examples of these. Evolution in present times, the key word here when you see evolution in present times is that you see, you think of natural selection. Okay, basically here we are showing, if we are looking at if we're looking at bacteria, we are saying that if we have a population of bacteria that are resistant, and we have some that were resistant and some are not resistant to an antibiotic. When we administer the antibiotic, the ones that are not resistant will die, the resistant ones will survive and they will reproduce. And then you'll carry on administering the antibiotic. And as you can see here, as you carry on administering the antibiotic, the red ones become more and the yellow ones become less, which means that over time, the population is going to become more resistant. And this is the reason why some drugs don't work anymore, because the population is becoming resistant to the antibiotic. Right, human evolution. Okay, possible questions for human evolution. You must know how to interpret a phylogenetic tree. Okay, you must know how to interpret a phylogenetic tree. Very important. Okay, you need to ask yourself when you are doing the phylogenetic tree, you need to identify who is the common ancestor, who is extinct, when did the species on the diagram originate, how long did they live for, and when did they become extinct. Okay, we're going to go through an example. Then you need to know the similarities between African apes and humans, so what we share with them, the differences using the diagrams of the skull, the foramen magnum, spinal cord, and the pelvis. Then we need to know the fossil, genetic, and cultural evidence that support this, and know the fossils that were found in South Africa. Know them. There are many, I know. At least know the ones that are found in South Africa and who found them and where they were found. And then lastly, the out of Africa hypothesis. Important here is exam guidelines. Straight from the exam guideline, that's what you need to know. Right, let's quickly look at the phylogenetic tree. Okay, we just have a technical issue quickly.
Excellent. Excellence is a habit. Excellence is a quality you have, a quality I have. It's a quality we have when we honor what is true to us. Yes, we are gifted differently. uNkulunkulu akasinganga ngokufana. Excellence is striving for your best. Whenever you have a particular project, test, homework, uzibuze, ask yourself, is this my best? Can I put in more effort? Excellence is striving to be in the top 1% of the area in which you are gifted. And so I challenge you, in every single thing that you do, in every single area that you're gifted in, whether it's sports, it's academic, ucula, whatever that gift is, strive for excellence.
Welcome back. Sorry about that, but we're right back on track. So, phylogenetic tree, very important. Also something that you can't really study for, but you need to practice the skill. Also, very good chance that you're going to see this. Okay, so as we said, we need to identify where the common ancestor is, right? But firstly, very important when you are looking at a phylogenetic tree is that you need to look at the timeline. Okay, you need to identify when the past is and when the present is. Okay, present will be at 0 and the past will be a number, so many million years ago. So the further back you go, that's where you'll find the common ancestor. In this instance, it's right there where the first branching takes place. That is the common ancestor, and then it branches off into the descendants. Okay, and then who is still in existence? Okay, which organisms are still in existence today? When you need to read that off, you need to see which line goes right to the present day or to 0. Okay, so in this instance, it is the Homo sapien. They are going right to the end. Okay, so they are still in existence. If they became extinct, then the line just basically ends in the middle of nowhere, like here. A. robustus became extinct. Okay, when did they appear? They first appeared when they had this branch, when there's a branching. So you have a descendant here. Okay, so here's the common ancestor. We have the first branching or speciation. And then we gave rise to the next one. So this is when the A. robustus appeared, if we're just looking at the A. robustus. Now, the question can ask you, when did A. robustus become extinct? You take your ruler. Right? You take your ruler, you put your ruler on your page, you draw a line from this end of where the line ends and you draw it all the way to the timeline. Remember, check how to read the timeline. In this instance, it's between 1 and 2. Okay? 1, this would be about 1.5, and then here we get 2. Okay, that's how you read it off. When did it first appear? You draw another line from there all the way down with your ruler. Okay, how long was it in existence for? You say, when did it appear? In this instance, it's 3 million years ago. And you subtract, when did it become extinct? 1.5 million years ago, and that's equals to 1.5 million years. Why am I just saying million years? It's because it was in existence for that amount of time. Okay? Right. Then we have the similarities between Homo sapiens and primates. Now, grade 12. I'm going to do this one in stages. I quickly want to bring up all of them because they are color coded. You need to look at your human, you need to look at your body. Okay, a good way to remember it is that look at yourself. Okay, look at yourself and then ask yourself, look at the different regions. Let's first look at the head. Okay, let's look at the head region. I've indicated them in blue. We have large skulls compared to their body mass. The eyes are in front. Okay, or we have binocular vision or stereoscopic vision. Okay, and then we are also having our we have our freely rotating arms. Okay, those are the ones that are indicated in purple. So you look at your own body, okay, and you use your arm in the exam room. Okay, we have freely rotating arms, right? We have long upper arms. We have elbow joints allowing rotation of the forearm. Okay, then it comes to the fingers. We have 5 fingers. We have opposable thumbs. Right, we have and then you can see, look at this picture over here of the African ape with the human. Okay, and you can see that we have opposable thumbs, and then we are also having bare fingertips with nails. Okay, we have 2 mammary glands. We have sexual dimorphism, which means that we have a male with male reproductive organs and a female with female reproductive organs. Okay, these are the similarities. And I've actually indicated it. There we go. We have indicated it with different colors. So you can actually look at the colors and try to remember the different things. But really look at your body. Okay, think about the head, what is similar, the arm, and which are the upper limb. Okay, your fingers, and then the mammary glands and the sexual dimorphism. Right, we are nearly there. Okay, then the differences between the humans and the African apes. Quickly, what I wanted to say, which was very important. When you need to tabulate the differences between the two, you have a table. Compare the same thing. So if they have to ask you the differences between the humans and the African apes with regards to bipedalism and quadrupedalism, okay, then you need to talk about, can you see they're comparing the foramen magnums, okay? So if you're going to have bipedalism here and you're going to have quadrupedalism here, you need to, the first point you need to talk about foramen magnum here, you talk about the foramen magnum there. You cannot talk about the foramen magnum here and here you're talking about the S-shaped spine, okay, or the C-shaped spine. So you need to compare the same things. Okay, so quickly, the important things here. For bipedalism, the foramen magnum is in a forward position. The spine is S-shaped. We have a shorter, wider pelvic girdle. In the African apes, with the foramen magnum is towards the back of the skull. It has a C-shaped spine which goes into the foramen magnum towards the back, and we have a longer, narrower pelvic girdle. In the humans, the cranium size is larger. In African apes, it's smaller. The palate shape is round in the human and rectangular in the African apes. The dentition, we have no gaps between our teeth. The African apes have diastema. They also have large canines and we don't. Quickly on prognathus. Prognathus is about this part, okay, the snout part that's protruding. So in African apes, they have prognathus because it's protruding here, whereas we have non-prognathus. That leads us to have a flat face, and they have a sloped face. And then we have the cranial ridge, that is this area here that's raised. We don't have it, but they have it. Okay, so then there's just the next one over here that shows you, just to show you that the spinal cord is entering over here. The foramen magnum is in a more forward position so that the spinal cord can enter vertically. This one we have a C-shaped spinal cord. The foramen magnum is towards the back so that it can enter there towards the back. Then in humans, this is the short and wide pelvis, and here we have the narrow and long pelvis. Right, fossil evidence. The main things here to understand is that in fossil evidence, we have transitional species, and these species are sharing characteristics of their ancestor and the characteristics of the species that will come next. In genetic evidence, we have mitochondrial DNA. Why do we use mitochondrial DNA? You get it, it's passed by the mother. And it's also an easier one, a more simpler form to do than nuclear DNA. And cultural evidence is tool making. That's what distinguishes us. So as they were busy evolving, some of them started developing tools, and the more complex the tools became, it shows us evidence that they were more evolved. Right, then these are just an example. You need to know all of these. You need to know Australopithecus africanus, Tahung Child, Mrs. Pless, and Littlefoot. You need to know who found them, where were they found. The same with Australopithecus sediba, which is also known as Karabo. Homo naledi. Quickly, Tahung Child is Raymond Dart, right? Mrs. Pless, Stykfontein Caves, Robert Broom. Littlefoot, Stykfontein Caves, Ron Clark. Karabo from Lee Berger and Brett Eloff, and they were in the caves near the Malapa. And then we have Homo naledi, found by Professor Lee Berger in Cradle of Humankind, and then the Homo sapiens, they were found in Limpopo, Free State, KZN, Western Cape, and by Prof. T.F. Dreyer. Lastly, the out of Africa hypothesis. Okay, so we have the out of Africa hypothesis. Mainly what you need to understand is that eventually as the organisms evolved and the species evolved, when they became Homo erectus, okay, and they were able to be fully bipedal, they actually moved out of Africa, okay, to other continents. What is the evidence of this? From your examination guideline, the fossil evidence is that the Ardipithecus fossils and the Australopithecus fossils were only found in Africa. Remember, Ardipithecus and Australopithecus came first. They were the previous ones. Okay, they were found only in Africa. The early homos, Homo habilis, for example, they were found in Africa only. But the early Homo erectus and the later Homo erectus and those were all found in Africa and in other parts of the world. Okay, that's how you explain it. And lastly, the genetic evidence, we look at mitochondrial DNA, the analysis of mutations in mitochondrial DNA. They trace the oldest female ancestor to East Africa. Right, lastly, right, that was a bit of a marathon, but please remember, always take your compass and your protractor, because you could get asked to draw a pie chart. Right? Have a ruler with numbers and segments on it so that you can measure things. Have a calculator that's working. Bring 2 different pens just in case the one jams. Have a pencil. Have an eraser. Okay, these are the things that you must prepare and take to the exam room. Grade 12s, good luck. I wish you everything of the best. Study hard. Be fully prepared. Make sure that you sleep well the night before. In the morning, you have a good breakfast, you get to school, you relax, you find a place, you get into your zone, and you relax, right? When you write your paper, make sure that you read your questions carefully, and everything will just hopefully flow towards you. And remember that the only place where where success comes before work is in the dictionary. Good luck for your exams, and good day.